Okay, I wanted to go over a quick video with you guys, what we've talked about the last couple of days in class. And really it's the last thing that we're going to be covering on the macromolecules, or sometimes we call those biomolecules. And there's four major types that are found in living systems. And there's a video that we watched in class. If I think about it, I'll put the link on there, but it's from the Amoeba Sisters, and it's really cool, and it kind of gives you some information to introduce the topic to you. But most of everything that's in that video I'll cover today, so if you don't have time, it's not that big a deal. Also, I want to let you know tomorrow we're going to be trying to squeeze in a benchmark test. So I will be posting that, be looking at that on your Google Classroom, and it will only help your grade. Okay, I will let it replace your lowest test score if it's better than your lowest test score. So be looking for that tomorrow and try to get that taken sometime tomorrow. So let's talk about these living systems here. The four major ones that are found in living systems, they are proteins, lipids, nucleic acids, and carbohydrates. So the last one here that we mentioned is the first one we're going to talk about. Carbohydrates is typically your body's preferred energy source. There's three main types that we will talk about. The first type is what we call a monosaccharide. This is kind of like a building block, okay? This is the simplest sugars. Glucose is a good example of that, as well as fructose. And these are the building blocks that will build more complex carbohydrates. So the second, if we add two of those monosaccharides together, we form what's called a disaccharide. Di meaning two. And the classic example is sucrose, which is made up of glucose and fructose. And then we have polysaccharides, poly being many. So there's many saccharides. Think about that. Many monosaccharides together will make a polysaccharide. And there's three main types of those that we typically like to talk about. Starch is one, which is found in plants, and glycogen, which are found in animals, and more specifically a lot of times in the liver of animals. Now, glycogen and starch are both what we call structural polysaccharides. That means they are both what we call storage polysaccharides. They are storage, not structural. They store energy for the plants and for the animal. And so it's this quick access for plants if the plant or the animal needs energy source. Starch and glycogen is there for them. Cellulose is a structural carbohydrate. There, I got it right this time. Structural carbohydrate. They're going to be found in the cell walls of plants, and their main use is going to be giving plants their shape and their structure. Because remember, plants don't have a skeletal system, so they have that cellulose to give them that support. It's also known as a more common term is fiber. So we can't digest cellulose. We don't have a ability to digest it. Now, it does serve a purpose for us. It kind of helps, think about fiber, it helps regulate your digestive system, keeping you regular and things like that, so you don't, get stopped up. But anyway, so it's beneficial for us along those lines, but we really can't digest it. And the reason being, real quick, we talked today about how now enzymes are not carbohydrates. They are actually proteins, but I wanted to show you this image here of how enzymes work. Enzymes have what's called an active site. So you'll notice here there is this active site. They're very specific to the substrate. So it's like a lock and key. So not just any substrate will fit into any enzyme. They're very specific to enzymes are very specific to whatever they need to break down, right? So for example, in your mouth, in your saliva, you have an enzyme called amylase, and it only breaks down carbohydrates. And so it starts the digestion of carbohydrates in your mouth while you're chewing your food. When you're chewing protein or fat or anything like that, and when you're eating that, the amylase doesn't work on it. It only works on the carbohydrates. So they're very specific. So what I wanted to show you, another cool thing about enzymes is after they break up a product, they can be reused. Okay, they can be reused again and again and again. They don't wear out after one use. They probably eventually would wear out, but they can be reused. But I wanted to show you this picture real quick. I'm not going to spend a lot of time on it because it won't be on your test or anything. But this is showing you the difference between starch and cellulose. So starch is made up of what we call alpha glucose monomers are, that's the monomer that we're dealing with, the building block, whereas cellulose is built of the beta glucose monomer conformation of this monosaccharide glucose. So if you'll notice here real quick, all of these OH groups here on the alpha glucose are on the bottom side. And on the beta, they're on the top side. So what that does is whenever we have starch, it makes all these hydroxyl groups on the bottom. Whereas on the cellulose, it alternates. It puts a hydroxyl group on every other up top, that hydroxyl group up top. What does that mean? It's complicated, but the main thing it means is we have enzymes in our body that can break this down, that fits like the lock and key. But we do not have enzymes. Those same enzymes that fit those will not fit cellulose. So therefore we cannot break those down. Let's move on to the next macromolecule, and those are lipids. Lipids are going to be your fats, oils, waxes, steroids. Remember, so they are what we call nonpolar. Nonpolar, make sure you don't get this confused. Nonpolar means that those carbons and hydrogens are sharing their electrons equally. Okay, so when there's an equal sharing of electrons, there's no pull one way or the other. So lipids are nonpolar. So that was why we talked about in the other video that if you put oil and water, it won't mix. It's because water is polar and lipids, fats, oils, things like that are nonpolar. So there's no attraction there between that polar water that has a charge, a partial charge, and lipids that don't have a charge. And so lipids are good for insulation. Think about animals that are marine mammals like whales and seals and things like that. They have that blubber that protects them. It keeps them warm, and that's what that fat does for them. It's also a good source of long-term energy storage. So if we needed to store energy for over the long term, fat does that. And sometimes what happens is, we eat carbohydrates, which is our body's preferred source of energy. But what happens is if we eat more than our body will burn and use, your body easily will convert that to fats for that longer term energy storage. And that's why sometimes people go on a low carb diet so that they don't convert all those excess carbs into fat. So when we talk about lipids, there are a couple of different types. There are saturated and unsaturated. Saturated lipids, if you'll notice the difference between these two, you'll see right here in this unsaturated, this is what we call a double bond. Okay, so between these two carbons there is a double bond, and because of that double bond right there, there are not hydrogens bonded here and here, because if you remember, carbon will only bond with 4 different things. So every one of these carbons is sharing 2 electrons with another element. So 2, 4, 6, 8, 2, 4, 6, 8, and even here 2, 4, 6, 8. There's that double bond counts as 2. So 2, 4, 6, 8. So there's not hydrogen. So we say that this is saturated with hydrogens, and this is unsaturated because there's no hydrogens right here because of that double bond. Hopefully that makes sense. I tried to go through it pretty quickly. Saturated fats are going to be animal source, and those are going to be like lard and butter and cream and grease, things that are solid at room temperature, okay? And they all have an animal source. Typically, they're worse for you than unsaturated fats overall. Now, unsaturated fats, if you think about these, they are going to be liquid at room temperature, and the source of these are plants. And these are more commonly, you think about vegetable oil, olive oil, canola oil, avocado oil, the peanut oil. Those are different things that are going to be liquid at room temperature. Not as bad for you as the saturated fats. Real quick, we talked in class today about another type of fat called trans fats. And what trans fats are, before we get into protein, trans fats are this, what they try to do, what food scientists try to do is take this unsaturated fat that is liquid at room temperature and make it to where it was solid. So for example, with peanut butter. Peanut butter, if you've ever bought natural peanut butter, you'll notice that it's got the peanut butter in it, but then there'll be this layer of oil, and you've got to stir it up. Well, what they try to do, food people try to do, is make that peanut butter just solid at all times, so it doesn't have that oil in it. And what they will do is they will hydrogenate, what they call partially hydrogenate those peanut oils, and they bombard this with hydrogen. And what it does is it turns those unsaturated fats. Instead of saturated, it makes them into trans fats, which we've discovered are worse for you than saturated fats. And so you'll see that sometimes in food that food contains partially hydrogenated vegetable oil or corn oil or something like that, canola oil, and typically they will make those trans fats, and those are worse for you than saturated fats. Real quick, proteins are the next thing we want to talk about. They're used for structure, for tissue, for defense in your immune system. Enzymes, which we talked about earlier, are a type of protein, and these are very important. Pretty much your whole body, if you look at yourself, your skin, your hair, your nails, your muscles, everything is made up of protein in your body. Very important. All the enzymes in your body, have thousands and thousands of enzymes that have jobs, and they're all made up of protein. The building blocks of proteins are amino acids. And so that's kind of what we call the monomer of a protein molecule, are these amino acids. So you put all these different amino acids together to make up a protein. And then, again, there's a picture of the enzymes, again, that are classic examples of proteins. The last major molecule that we want to look at are nucleic acids. Nucleic acids are a biomolecule that stores cellular information in the form of a code. There's there's only two types of nucleic acids, and those you'll be familiar with: DNA and RNA. And again, these are the building blocks for nucleic acids are going to be these things called nucleotides. Okay, nucleotides are the building blocks. And if we look at a nucleotide in this picture here, this is a nucleotide. It consists of a phosphate group, a sugar, and then what we call a base, or a nitrogenous base. Now, the phosphate group and the sugar just make up the backbone, kind of just the support of that DNA molecule. So if you think about a ladder, they would be like the sides of the ladder. But in the middle is where all the information is. Those rungs of the ladder is where the information is. And these are called bases, or nitrogenous bases, base pairs. And you'll notice here, A's bond with T's, G's bond with C's, and T's with A, and C with G. The A stands for adenine, T thymine, C cytosine, and G guanine. So the main thing you would need to know, we'll talk way more about this later on in the year, is that with these are what contain all your information, your genetic information, are all with these letters. And so you've probably heard of these letters before: A, G, C's, and T's, and things like that. But we will, again, talk about those later. So that's a quick run through on these macromolecules. Again, if you wanted to check out one of those videos, there's some videos out there that are kind of informational as well. So be looking for that benchmark tomorrow. And if you guys, other than that, you have a good day. I'll talk to you later.